Hello everyone. Thank you for joining our webinar today to discuss predicting disease in biofluids and tissues with Raman Spectroscopy. I'm Carrie Burton, your Renishaw host for today's webinar, and with me today is Jenny Ferguson and Dale Borman. Before we begin, please note that you are on mute and will remain in listen-only mode throughout the webinar. We encourage you to submit your questions or comments at any time during the session using the control panel on the right side. We will be answering as many questions as possible at the end of the presentation, and we are recording today's webinar and will have it available for on-demand viewing. And now I will hand it over to Jenny Ferguson, Raman Application Scientist at Renishaw PLC.
Hello everyone and welcome to the webinar. Today we are going to be discussing predicting disease in biofluids and tissue with Raman spectroscopy. So to begin with, I'm just going to give it a brief overview of exactly what we're going to be discussing today. And this will start with an introduction into the application of Raman in biology. I will discuss the type of information you can get out of spectra and some of the advantages of Raman compared with other conventional analysis methods. I will then highlight how Renishaw's Raman systems have solutions to the many challenges that you might face when analysing biological samples. And I will then discuss how we can use Raman for identification of tissues and biofluids and the instruments that Renishaw have that will enable you to do this in an easy, efficient way. Finally, I will take you through a standard workflow for using Raman for detecting disease in biofluids and tissues. After this, I will pass over to my colleague Dale, who is going to talk through some specific case studies which demonstrate the classification of tissue and biofluids using Renishaw's Raman systems. So let's get started with an introduction into Raman spectroscopy of biological samples. Raman spectroscopy is an extremely powerful technique that can be used for chemical characterization and identification of samples. It works on the basis that a laser light radiates a sample, interacting with a molecular bond which cause a very small proportion of the light to undergo Raman scattering. This Raman scattered light is measured and provides us with a spectral fingerprint specific to that material. When applied to biological samples, we can use this spectrum to determine the presence or absence of constituents in a sample. Such information can ultimately allow us to determine the state of cells, tissues, and biofluids, and more specifically, we can use this to determine if they are normal or diseased. Renishaw have a range of Raman systems suitable for analysis of a variety of biological samples. So what type of information do we get out of a Raman spectrum? A Raman spectrum of a biological sample contains lots of information. This is an example Raman spectrum of a biosample, and it indicates the different biomolecules the bands are representative of. It also shows the different types of studies that are possible using Raman spectroscopy. For example, the bands in the 1400 to the 1800 wave number ranges are representative of lipid and proteins, and they can be used for lipidomic and proteomic studies. Raman spectra can provide lots of different information, such as the presence or absence of specific biomarkers that can allow detection of abnormalities, diseases, and cancers, the presence and structure of proteins, changes in nucleic acid and metabolic studies, and investigation of lipid saturation and composition changes. All of this information can provide us with essential information about the state of cells, tissues, and biofluids. We obtain this information by comparing variations in the Raman bands between and within different samples. We can use Raman spectra to build up a Raman image of samples. For example, here we are showing the Raman image of a rat colon. This image is a representation of thousands of Raman spectra, and large datasets such as this are really common when using Raman, and so applying multivariate analysis, which allows us to reduce the dimensionality of datasets, is really commonplace. In this example, principal component analysis has been performed on the data to allow us to more easily identify the presence of the biological components in this colon. Each of the graphs that we can see here around the outside of the image are loadings, which allow us to determine the spectral features which vary across the samples. In this case, it allows us to identify the different biomolecules present in the sample. Within this Raman image, we can see there are 4 different principal components corresponding to muscle layers, submucosa, goblet cells, and DNA amplifying cells. As we can see, the spectra for each of these groups are slightly different, and this allows us to separate them when applying principal component analysis. Peaks that we can see are labelled and they correspond to certain biomolecules, and whole spectra represent the combined chemical makeup of that particular tissue. For example, we can see that the purple areas are representative of muscle and green are goblet cells. The high resolution which can be achieved with Raman imaging enables tissue structures to be accurately labelled. The application of multivariate data analysis techniques such as principal component analysis and linear discriminant analysis are methods which are commonly used when performing classification of disease in different samples. And we will see this applied later on in this way using the Renishaw data classifier. So aside from the information that we can obtain from Raman spectra, what are the other advantages of using Raman spectroscopy compared with other conventional analysis methods? Well, firstly, Raman can be used non-invasively and non-destructively. And this is extremely advantageous when you're looking at biological samples, which can be extremely sensitive to damage. Secondly, Raman is label-free, and this eliminates the possibility of any artifacts resulting from the need to fix or label samples for analysis. It also means that minimal preparation time is needed prior to performing Raman on samples. Raman can even be performed on living cells and animals. This is dissimilar to other biomolecular techniques, which often require significant preparation. Later on in the presentation, we will discuss sample preparations of tissues and biofluids more thoroughly, and the best ways to prepare your samples for undergoing Raman analysis. Another advantage of using Raman is that spectra can be collected in a matter of seconds, and when using Renishaw's streamlined imaging technique, thousands of spectra can be acquired in a couple of minutes. The laser power at the sample and the measurement conditions are really easy to control on Renishaw's software, and this means that the measurement conditions are really easy to optimize and can be performed rapidly. Ultimately, the fact that Raman provides you with a full chemical fingerprint of the sample, allowing for identification of particular constituents and biomolecules, and its ability to do this in a non-invasive and non-destructive, label-free way really does make Raman an excellent tool for analysis of biological samples. So let's look a little bit more closely at the different types of biological applications of Raman and different sample types that can be analysed. The applications of Raman in biology are extremely widespread, as you can see from this flowchart. And each of these different branches represents a different application of Raman in biology. For example, Raman can be applied to reproductive biology or virology. It can also be applied to biofuel and drug development, including studying drug delivery in vivo and in vitro. It can be used for various omics analysis, as we briefly discussed earlier. And the application of Raman that we're most interested today is the ability of Raman to be applied for disease detection and medical diagnostics. So let's look a little bit closer at these specific applications. So specifically here, we have two main branches coming off of our medical diagnostics and disease detection section. Ex vivo involves the analysis of tissues and biofluids which are not in situ for disease detection. So for example, tissue which has been removed from a patient during a biopsy for detection of cancer. The samples are then taken away to a lab or a clinic and analysed on an instrument. On the other hand, we have in vivo analysis, which is ultimately using a probe in situ to collect Raman spectra and determine if cells, tissues and biofluids are diseased or normal. So let's discuss in vivo and ex vivo in a little bit more detail. The benefit of ex vivo work is that there is no time constraints, and so it is really useful for performing method development in order to ensure that you're collecting the most optimal data. That means that measurement conditions can be optimized to ensure that they are giving you the best signal to noise ratio possible in the minimum time necessary, and that no damage is being done to the cells or the biofluids during data collection. The results obtained ex vivo can then be used to build, optimize and validate models for disease classification, and this ultimately makes ex vivo the natural first step of sample analysis before performing in vivo analysis. In vivo measurements are when data is collected in situ, meaning there is no need for tissue or sample removal. When data is collected, a model can then be applied to rapidly classify sample types, and this model will be built with the ex vivo results of known pathology. This type of analysis could ultimately lead to real-time detection of diseases. However, we're a little way off that just yet. Renishaw has research-grade systems for both in vivo and ex vivo analysis, and while these systems are not suitable for use in diagnostic procedures, they can be used for disease detection within research settings. So let's now look more at the ability of Renishaw's Raman systems to predict disease in tissues and biofluids. Let's first discuss some of the main challenges that are faced when we analyse biological samples. The first difficulty is the ease and ability to classify tissues and biofluids, and this ultimately breaks down into three main steps. The first of these is the need to identify the correct area of the sample, and this is mainly to do with sample preparation and the best way to go about analysing your tissues and biofluids. Knowing how to prepare your sample for analysis can ultimately produce the best Raman results, and in the next few slides I will provide some tips on how we do this at Renishaw. When it comes to tissue samples, identification of the correct area of the sample can be difficult, and this is mainly due to the fact that Raman is not compatible with stained tissue sections as it causes fluorescence in the Raman spectra. Fortunately, Renishaw have an image alignment tool that allows you to overlay your stained tissue section with an unstained section to identify areas of interest for data collection. The next challenge that is often faced is the need for appropriate instrumentation. There is a need to perform both in vivo and ex vivo measurements and also have instrumentation that is suitable for research and in a clinic. As biological samples are weak Raman scatterers, the time to collect Raman spectra can often be time consuming, and so the instrumentation must have high performance. The last cause for concern is the transferability results, and that is the ability to collect the same results across different systems in different locations. Bringing Raman to the clinic is a challenge in itself. The ultimate aim as researchers working in disease detection is that this can be applied directly to the patient for automatic disease detection. Bringing Raman to the clinic would facilitate rapid analysis of tissue and biofluid samples without the need to send it off to the lab. It would allow real-time information to be generated and allow for faster disease detection during biopsies or surgeries. This would provide additional support for early disease detection. However, for a system to be compatible with a clinical environment, it would need to have a small footprint, be easy to clean, while still maintaining high performance and reproducible results. So what's the solution? Well, we need instrumentation that supports efficient disease classification in research and clinic-based settings, and Renishaw have the perfect systems for just that. This is the Renishaw R816 biological analyzer. It's a compact benchtop Raman imaging system for tissues and biofluid analysis. Its compact design makes it ideal for small lab spaces and clinics, and it is extremely easy to use, making it ideal for users from all background levels. This system has been designed exclusively for the biological and clinical community. It combines the high performance needed for research and incorporates the ease of use, small footprint, and minimal maintenance needed for the system to be ideal in a clinical setting. This system is designed for ex vivo measurements of tissues and biofluids and comes with sampling accessories that makes it really easy for measurements to be performed on both. Next, we have the Renishaw Virsa in vivo biological analyzer, which enables the study of biological samples in situ. It is compact and portable, meaning it can be moved between multiple sites with ease for measurements to be performed at multiple locations. Its internal components are fixed, so the signal is unaffected by movement of the fibre. Its compact configuration makes it easy to use, while its high performance makes it an optimal candidate for use in disease detection. All of its materials are biocompatible and sterilizable, making it ideal for use on biological samples. So how do we go about using these systems for disease detection? Well, I'm now going to walk you through the workflow which will demonstrate the 3 steps which will allow us to classify Raman data and determine if tissues or biofluids are diseased or not. The first step is sample preparation and the collection of Raman spectra from the biofluid or tissue. Secondly, we use Renishaw's data classifier to build and validate a classification model with known pathology. And finally, we can apply this to new samples with unknown pathology and ultimately determine if they are diseased, abnormal, or normal. Let's begin with step 1 and discuss this in a little bit more detail. And first we'll begin by looking at the sample preparation of biofluids. The sample preparation for liquids vary. For instance, for blood, it is usually centrifuged for analysis of the constituents. The R816 biological analyzer comes with sample and accessories which are optimal for use with fluids, gels and powders. For instance, we have a welled steel slide which has 3 wells of different depths, allowing you to analyse different volumes of liquid. Steel slides are optimal for use with Raman as they have no Raman background and they're also cheaper than the commonly used calcium fluoride slides. They're much more beneficial than using glass slides as they don't have any Raman background which interferes with the Raman signal, and it means it doesn't require any background subtraction, such as the case with glass slides. The R816 also comes with different stage inserts, such as this one shown, which is adapted to fit well plates. This combined with the well plate mapping software that is available on Renishaw Raman systems makes measuring biofluids really straightforward. So next, looking at the sample preparation for tissues. First, we will discuss bulk tissue, and the step 1 for this involves preparing the bulk tissue for analysis and collection of the Raman spectra. So how do we go about doing this? Prior to analysing bulk tissue, you need to ensure that the tissue section is clean of any contaminants. This is particularly relevant for blood, which could interfere with the Raman signal. Data collection can then be performed using the Renishaw Virsa in vivo biological analyser or the R816 benchtop biological analyser. The R816 also comes with a bulk tissue holder. It is enclosed with a calcium fluoride window through which measurements can then be taken. This accessory is ideal for bulk tissue measurements as it ensures the tissue does not dry out during measurements and data collection. Looking now at the thin tissue section, the first consideration when analysing thin tissue sections is the identification of the correct area of the sample for analysis. Areas of interest are normally identified by staining tissue samples. However, as previously mentioned, this causes fluorescence in the Raman spectra. Renishaw have an image alignment tool that allows you to take a snap of a stained and unstained tissue section and overlay them, easily identifying the area of the sample of interest. Data collection can then be performed using the biological analyzer R816. So now let's look at step 2, and this is to build and validate the classification models. This is done using the Renishaw data classifier software. This software does not require any knowledge of model building or classification techniques. It requires a user to input the spectra of known tissue types, such as cancerous and non-cancerous, or of diseased and normal biofluids to build the model. It is able to process multiple spectral datasets simultaneously and has a function for validating the dataset so the user can ensure it is working properly prior to use. The algorithms used in the data classifier actually take into consideration any instrumentation variations and have algorithms to automatically deal with these to ensure that samples are not misclassified on this basis. The data classifier is based on PCI LDI, which we discussed previously, and that is a multivariate data analysis technique. The images here show the model building tool in the data classifier, which provides information on the model sensitivity and specificity. The graph shown is one of the loadings, and on this there are spectral features that can be identified which have been used by the model to classify the different data sets. And below this we can see the histogram which demonstrates the separation of the model building data showing the diseased and normal spectra. Once a model has been built in the data classifier tool, it can then be saved and applied to unknown samples. So once we have built our model, we can move on to step 3, which is classification using Renishaw's data classifier. And this is when we apply our model to tissues and biofluids of unknown pathology. So we can apply this to the spectra obtained from our tissues or our biofluids to determine if they are diseased, abnormal, or normal. And here we can see PCI and LDI results for 2 types of cancerous tissue shown in red and blue, and the separation from normal tissue which is shown in green. Again, we can see the loadings just above these plots. And on these loadings, we can see spectral variations between the 3 pathologies. So ultimately, that's the 3 main steps. First off, sample preparation and data collection. Second, build and validate our model. And third, apply this model to unknown sample types. This simple workflow can be applied to any sample types using data collected from the biological analyzer R816 and the Virsa in vivo biological analyzer. The next thing that I want to discuss is the transferability of results between instruments. A study was performed by Renishaw in collaboration with the University of Exeter, University College London, and the Gloucestershire Hospitals. The aim of the study was to investigate the ability of the Renishaw data classifier to detect different tissue types on 3 RA816 biological analyzers in 3 different locations. The data from the 3 instruments was successfully separated using the Renishaw data classifier with high sensitivity and specificity as shown in the table. We can also see some of the spectra here of the 2 different disease stages. There are some clear differences in the 2, such as a reduction in the intensity of the band at 1262 wave numbers. So from this study, we can be certain that the Renishaw data classifier and the RA816 biological analyzer is able to differentiate different tissue types with high sensitivity and specificity across different instruments. We can therefore be sure of the reliability of the data and results obtained using this equipment. I'm now going to hand over to my colleague Dale, and he is going to run you through some specific case studies where we have utilised Renishaw Raman equipment for detection of disease in biofluids and tissue samples.
For the second part of this webinar, we'll discuss some case studies which have used the workflows outlined in part 1 in order to study both tissue and biofluid samples. The first example looks at classification of brain tumours from bulk tissue biopsies. This work was undertaken with Oxford University and the John Radcliffe Hospitals and has been published in the Journal of Neurosurgery and Neuro-Oncology Advances. Before we go into the details of the study, here is a short video from James Livermore, the researcher behind the work. In this video, James describes how he has used Raman spectroscopy to understand the key characteristics of brain tumour tissue. He explains how the generated information could benefit cancer treatment strategies.
I'm currently doing a PhD in applying Raman spectroscopy to brain tumour research, and we're using the Renishaw biological analyser to try to learn a bit more about brain tumours. So it's become increasingly important to know the genetic subtyping of these tumours actually at the time of surgery to plan treatment, not only surgical treatment, but then later on radiotherapy and chemotherapy. The beauty of Raman is that you don't need any sample preparation at all. The samples can be put straight in the analyser and it can give you an answer pretty much straight away.
Brain glioma is the most common and most aggressive primary tumour of the central nervous system. Surgical removal of tumour tissue is important in the management of patients with glioma. Increasing evidence is showing that the more tumour tissue that gets removed, the better the patient's prognosis of progression-free survival. However, given that we are working in the brain, removal of any healthy tissue will have serious health implications for the patient. Removal of non-cancerous tissue can cause significant neurological damage, and so it is extremely important that the surgical removal of cancerous tissue does not come at the cost of disability. An added complication is caused because of the diffuse nature of gliomas. Cancer cells can break away from the main body of the tumour and infiltrate the surrounding brain tissue. This makes maximising tumour removal challenging and means that complete tumour removal is not possible. Raman spectroscopy may provide an effective method for accurately separating healthy and cancerous tissue. The high specificity which Raman offers at the molecular level means that very small chemical changes in tissue can be detected. This has the potential for identifying tissue with infiltrating tumour cells and not just bulk tumour tissue. Importantly, as Raman does not require extensive sample preparation, analysis can be performed in fresh tissue samples. A sample workflow was first established for analysis of biopsy tissue by Raman spectroscopy. Biopsy samples were surgically removed from patients with and without brain tumours. The biopsies were first separated into 2 equivalent parts. The first part was mounted on a mirrored stainless steel slide and underwent Raman analysis, whilst immunohistochemistry was performed on the second part to verify the generated Raman data. Raman spectra were collected using a Renishaw biological analyser, which was stationed within the clinical laboratory. In total, 9,799 spectra were collected from fresh glioma tissue, and these spectra were collected from 62 different patients. 1,825 spectra were collected from fresh, non-cancerous brain tissue, and these came from 11 different patients. The spectra were then used to build classification models, which could be used to characterize fresh brain tissue and assess for the presence of glioma. Using the collected spectra, a classification model was built to separate tumour and healthy tissue. Separation between the 2 groups could be achieved due to the very small differences in the Raman spectra. Here we can see the average spectrum for each of the classification groups within the model. Both spectra show typical biological peaks. These correspond to proteins, lipids, and DNA, but variations in the relative intensities of these peaks allow them to be separated. These small spectral differences are represented more clearly in the different spectrum shown below. A PCA-fed LDA model was used to separate the two groups. This used a combination of principal component analysis and linear discriminant analysis. From the generated principal components, those which represented a significant difference in the data were identified by analysis of variance, and these were then grouped using LDA. Verification with equivalent stain samples showed that the generated model could predict cancerous or healthy tissue with high sensitivity, specificity, and accuracy. The clear separation of the two groups can be seen in the histogram on the right-hand side. This model could then be applied to unknown tissue samples to assess for the presence of glioma. Another consideration for neurosurgeons is the genetic subtype of the glioma. This can determine how well a patient might respond to surgical removal of tumour tissue and the likelihood of the cancer returning. The most common and most clinically relevant genetic subtypes of glioma involve mutations of the isocitrate dehydrogenase gene, or IDH. Mutation of IDH are currently detected by immunohistochemistry and genetic sequencing. However, Raman spectroscopy may offer an effective alternative to identifying the IDH mutations. The previously used glioma sample sets contained a combination of IDH wild-type and IDH mutant tissue. Thus, a second classification model could be built to separate the two genetic subtypes. We can again see the average spectra for each of the classification groups. Although the spectral differences between these two groups were not as clear as those between tumour and healthy tissue, they were still enough to allow a model to be built which could separate the genetic subtypes. A PCA-fed LDA model was once again used to distinguish between the two groups. Verification with equivalent stained and sequenced samples showed that the generated model could predict IDH mutations with high sensitivity, specificity, and accuracy. Separation of the two groups can again be seen in the histogram on the right-hand side, and the associated ROC curves show high model performance. This model could therefore be applied to glioma tissue to assess its genetic subtype. Once classification models have been built, unknown tissue can be classified rapidly. A typical workflow for this would involve extraction of biopsy tissue, mounting the tissue on a grooved stainless steel slide, placing the slide in the spectrometer and collecting between 150 and 200 spectra, and then applying the classification model to characterize the tissue. By using the Renishaw biological analyzer, spectra could be collected very rapidly, and tissue samples could be classified in under 15 minutes. This rapid turnaround from biopsy removal to tissue classification offers the potential for real-time data generation. This could provide additional information to help surgeons identify tumour tissue and maximize their resection strategies. The generated Raman data could also complement more conventional biomolecular techniques. To put the potential of Raman spectroscopy into context, we compared its performance with 5-aminolevulinic acid or 5-ALA-induced fluorescence. 5-ALA-induced fluorescence-guided surgery is the current gold standard method for detecting brain glioma. It involves patients ingesting a dye which causes the tumour to fluoresce. However, the technique suffers from poor sensitivity beyond the visible tumour margin. It can resolve bulk tumour very well, but cannot detect tissue with infiltrating cancer cells. Raman spectroscopy, on the other hand, is much more effective at identifying the affected tissue, and hence showed a much higher sensitivity and accuracy when the tumour versus healthy model was applied. Thus, it is feasible that the two techniques may be able to complement one another to provide highly accurate detection of brain glioma. In this first case study, two classification models were built, one to separate healthy and cancerous tissue, the other to separate two different genetic subtypes of glioma. Both of these models demonstrated high sensitivity, specificity, and accuracy, indicating their potential for characterizing unknown brain tissue. Once models have been built, the whole process from biopsy removal to classification could be performed in under 15 minutes. This could allow real-time generation of information, which may be useful for surgeons when devising surgical strategies. The second example looks at ovarian cancer detection in biofluids. This work was performed by researchers at the University of Central Lancashire and the Lancashire Teaching Hospitals and is published in Analytical and Bioanalytical Chemistry. Ovarian cancer is currently the 7th most common cancer in women worldwide. As the associated symptoms are vague, most cases are diagnosed too late when the cancer has already metastasized. There is therefore a need for earlier disease detection. Current diagnostic approaches to detect the disease include clinical examination, imaging techniques such as ultrasound or CT scans, measurements of antigen levels in serum, or analysis of tissue biopsies. However, each of these techniques are invasive, expensive, and time-consuming. An alternative approach may be to use Raman spectroscopy to analyse molecular markers in biofluids. Removal of a liquid biopsy from a patient is much less invasive than taking a tissue biopsy, and minimal preparation of the biofluid sample is required. Additionally, the nature of Raman analysis lends itself to automation of processes. Raman spectra were collected from 3 different biofluids: blood plasma, serum, and ascitic fluid. For each of the biofluids, 6,210 spectra were collected from 18 different ovarian cancer patients, and 6,900 spectra were collected from 20 different patients with benign gynecological conditions. The spectra were then used to build classification models to test for ovarian cancer. Verification of models and assessments of their performance were provided by histological analysis of equivalent biofluid samples. PCA-LDA models were built for each of the biofluids to separate ovarian cancer and benign controls. Here we can see the average raw spectra for cancer and control classification groups for each of the biofluids. Differences between the cancer and control spectra can be seen more clearly in the spectra below, which have undergone normalization via the extended multiplicative scatter correction method and baseline subtraction. The scatter plots illustrate the separation of the 2 classification groups by the PCA-LDA model. Of the 3 biofluids investigated, the model built using spectra from ascitic fluid performed the best. Verification with histological analysis showed that the model could predict cancer with high sensitivity, specificity, and accuracy. This model could therefore be applied to ascitic fluid from patients to assess for the presence of ovarian cancer. As Raman spectroscopy provides a biochemical fingerprint, we can not only detect cancer, but also begin to understand the associated biochemical changes that are taking place. If we look at the difference between spectra from ascitic fluid from patients with and without cancer, we can see that the main differences involve peaks corresponding to protein and carbon-hydrogen chains. Taken together, these spectral differences indicate a change in collagen composition in the ascitic fluid when cancer is present. Thus, Raman spectroscopy not only allows models to be built in order to separate healthy and diseased biofluids, but it also provides a wealth of information to allow us to understand the chemical changes which underpin the classification that's taking place. In the second case study, a PCA-LDA classification model was built to separate biofluids with and without ovarian cancer. Separate models were built for blood plasma, serum, and ascitic fluid, and the model built using ascitic fluid performed the best, achieving a high sensitivity, specificity, and accuracy. The results indicate the potential for using the model to detect cancer from liquid biopsies taken from patients. Once the model has been built, tissue classification can be performed rapidly and thus could lead to earlier detection of ovarian cancer. In part 1, we introduced our Virsa in vivo biological analyzer for in vivo analysis of biological samples. In this case study, we demonstrate the ability of the system to distinguish between different tissue types. In vivo analysis does not require removal of tissue and therefore can be less invasive than ex vivo techniques. The ability to analyze tissue in situ also speeds up the generation of information. The probe-based system gives high-resolution chemical spectra, which are comparable to spectra collected using non-probe-based systems. These spectra can be used to build classification models to separate different tissue types. We first assessed the ability of the system to separate tissue from different animals. Spectra were collected from bulk tissue, and in total, 40 spectra were collected from beef muscle, 70 spectra were collected from chicken breast, 40 spectra were collected from pork muscle, and 40 spectra were collected from chicken cartilage. Spectral differences were apparent in the collected spectra from each animal type, as can be seen in the average spectra shown. These spectral differences allowed a PCA-LDA model to be built using Renishaw's data classifier software. The resulting scatter plot showed clear separation of the different classification groups. This data indicates the potential for using the Virsa in vivo biological analyzer for studying different tissues and assessing biomolecular changes with disease. To provide a more representative example of what may be required when studying tissue in situ, we use the system to analyze different tissues from the same animal. 70 spectra were collected from bulk chicken breast tissue, 40 spectra were collected from bulk chicken cartilage, and 50 spectra were collected from bulk chicken thigh tissue. Spectral differences could be seen in the collected spectra from each tissue type, as can be seen in the average spectra shown. A PCA-LDA model was once again built, and the resulting scatter plots showed good separation of the different tissue types. In this case study, we demonstrated that our Virsa in vivo biological analyzer is able to analyze bulk tissue effectively. The generated spectra showed high resolution, which allowed small differences to be resolved. This enabled classification models to be built which could separate different tissue types either from different animals or from the same animal. The data demonstrates the potential for using our Virsa in vivo biological analyzer to study tissue in situ. This will allow classification models to be rapidly applied to unknown tissue without having to remove a biopsy, and valuable information about tissue state can be obtained very quickly. Within this webinar, we've discussed the potential that Raman spectroscopy is showing within the field of medical research and diagnostics. Tissue and biofluid samples can be analysed in a completely unaltered form with no requirement for labelling. The technique is therefore well suited to the analysis of fresh tissue or fluid taken directly from the clinic or operating theatre. With Raman spectroscopy, samples can be analysed very rapidly. This is aided further by Renishaw's hardware design, which is helping to bring Raman spectroscopy into a clinical environment. The biological systems are compact and easy to use for non-Raman specialists, with automation of procedures where possible. In addition, in vivo analysis is supported with the Virsa in vivo biological analyzer, which further reduces the invasiveness of the analysis. With the potential for rapid collection of large numbers of spectra, accurate, sensitive and specific models can be built to assess for disease in tissue and biofluid. Once built, classification models can provide valuable information about the sample very quickly. This could be very useful for initial screening of a sample and may provide good support for conventional bioanalytical techniques. Finally, both hardware and software have been developed with reproducibility and transferability in mind. If models can be transferred between sites and they consistently perform effectively, this will facilitate larger scale studies and allow general workflows to be developed and adopted as standard procedures. At this stage, I would also like to reinforce that the Renishaw RA816 biological analyzer and the Virsa in vivo biological analyzer are designed for research purposes only, and whilst they can provide useful information to aid in the detection of disease, they should not be used as a routine diagnostic approach. With that, I would like to close the webinar and welcome any questions.
Thank you, Jenny and Dale. So now I invite you two on camera for our Q&A session. And a reminder to the attendees that to submit their questions using the control panel on the right side. And if we don't get to you today, we'll have someone follow up with you later. So hi, Jenny. Hi, Dale. So I'll pass it over to you, Jenny.
Thanks very much, Carrie. Okay, so we'll get started with our first question then. So in the case studies, in case study 1, the spectra were really similar and they managed to get an accuracy of 90% in the model for the IDH mutations. However, in case 2, the cancer spectra were relatively different, but they only got an 80% accuracy. How is it that happens?
So I think the first thing that we need to consider is that the spectra that we see vary depending on the disease. So I don't think we can specifically say the spectra looks similar, therefore we would expect lower model performance. Ultimately, it comes down to the variation within groups as well. So it may be that even spectra belonging to the same group show more variation, and therefore a bigger difference may be required to actually separate these spectra. So with the brain spectra, even though they were very similar, it may be that the spectra within the specific groups are even more similar. The other thing to note is that the spectra which were shown in the second case study, which obviously looked a bit more different, had had a baseline subtraction applied. So this is another process and procedure that we can take, which highlights kind of the differences between these spectra.
Okay, perfect, thank you. So in slide 6, there was a scan shown of a tissue section. How long did it take for this scan?
So this image actually showed the results of principal component analysis. So it specifically showed us the different PC loading, the different principal component loadings for that analysis. Generally, we have quite a lot of control over our scan times, and we can control this depending on the resolution that we'd like to get out. So we can actually image tissue samples with sub-second acquisition times and still generate strong Raman spectra. But then if we want to perform with a very high spectral resolution and a high spatial resolution, we can increase this acquisition time to kind of accommodate for this. We have several technologies and approaches as well, which can reduce the acquisition time further. So one of these techniques is Streamline. So this is a technology that Renishaw has developed, which uses a laser line instead of a laser spot, and this means that we can collect from different spatial regions simultaneously to improve our throughput and reduce our acquisition time.
Yeah, and I think that the really good thing about Streamline as well is that because it uses a laser line, it means that the power of the laser is distributed across this line, and that's particularly beneficial for biological samples in case there's any sort of sample damage and that sort of thing. It sort of mitigates that additional risk. Perfect, thank you. So the next question is the software integrated into the system?
So we do offer the software, the data classifier software with our R8816 systems, and this is generally because the whole kind of package has been designed for ease of use. So it's designed for non-Raman specialists to be able to effectively analyze their data, and therefore we generally advise that the two are used together, but it is possible to perform your own analysis as well from data collected with the system, but generally we advise that it's all used as a whole package.
Yeah, because it's a really nice workflow, as we showed. There's just sort of 3 steps where you do your data collection, your sample preparation, then your data collection, and then move on to your data classifier. And it's really useful because whenever you build your model with the data classifier, you can just reapply that to different samples. Perfect. So what spectral resolution has been used for this work?
So both the R8816 system and the in vivo probe system both have a spectral resolution of 2.5 wave numbers. So generally for any kind of biological sample that we may want to look at, this is more than enough spatial resolution in terms of the peaks that we want to identify within our samples.
Thank you. So what is special about the performance for using wells made of calcium fluoride? So
generally, the reason why people use calcium fluoride is because of the background that or the fact that the background it generates is a lot easier to deal with than other types of substrate, whether that's glass or quartz. Generally with Raman, because all the work that we've done has been with a 785 laser, or generally that's what we advise for tissue work, but this generates a strong interfering signal from glass, and therefore we advise using substrates such as calcium fluoride because they give a sharp peak which can easily be removed from spectra to kind of prevent this interference. We also provide stainless steel mirror finish slides. So we've actually generated data which show that these are an even better and a lot cheaper approach to actually mounting different samples. And this is because it doesn't generate any background at all.
Yeah, so it's really useful, particularly for whenever you're processing your data afterwards, that you don't have to do any sort of subtraction of the background from it. So it can really speed up your whole process. Perfect. So our next question is, how do you decide where to focus in blood serum?
Yeah, so this is an interesting question. And I guess it means that when we analyze something like a cell or a biofluid, the approaches that we take are quite different. So if we're imaging a cell, for example, we would want a high confocality, whereas if we were looking in a biofluid sample, this becomes less important. And what we really want to get is an average representative spectrum of that biofluid. So what we may do is use a lower magnification objective in order to maximize the Raman signal that we get out. And this, because it's kind of coming from a larger focal area, will give us a more representative average spectrum of our biofluid.
Perfect, thank you. How precisely can a model determine the type of cancer disease by Raman spectra in the case of an unknown sample?
So the models that we've shown are very precise. As we can see, the spectra are obviously very similar, and yet we can still accurately and precisely separate these two spectra.
Yeah,
but,
that really comes down to as well using like PCL, LDI. That can be quite useful in this case in terms of the classification technique. So whenever we use PA, we use that to classify, and then we can further put these into groups based on the LDA whenever that's performed.
Yeah, so I think, yeah, exactly. And I think if we're talking about unknown samples, that's kind of the overall aim of this approach that we take, is to generate these models which can accurately separate spectra, and then we apply that to unknown samples. And we have a variety of validation techniques that we can apply just to make sure that our models are working really well before we actually apply them to our unknown samples. So we can use known samples to provide validation of our models, but we also get a lot of outputs, things like ROC curves, to actually assess how reliable our models are and how well they perform. And therefore, when we do apply these to unknown spectra, we can be sure about the accuracy and how reliable they actually are.
Yeah, we know that we can be absolutely certain of what we're getting out of it. Perfect, thank you. So we only have time for one final question. So can you use map data and single spectra when you're using the data classifier?
Yes, you can. There are two kind of ways that we can collect our data. So the idea behind model building is that we collect large volumes of spectral data. So we can either take point spectra, whereby we collect from specific regions which we manually select in our sample, or we can actually take a map in terms of our data collection. And this is a faster way and perhaps a more supervised way, a more unsupervised way of actually collecting our data. And then at the other end of the model building process, we can then apply our built model to map data as well. And this will assign each of the pixels to one of our different classification groups. So this is a nice way that we can, for example, identify something like a tumour boundary.
Yeah, so something to keep in mind if you want to make your data collection a little bit more automated. Perfect, thank you very much. I'm going to hand back over to Carrie now.
All right, thank you everyone for joining us today. Please visit our website for on-demand viewing of our previous webinars and be on the lookout for information on our next webinar in March. Also, please scan the QR code or visit the website shown to provide feedback on our webinar, where you can also ask a question or book a discussion with one of our application scientists. So thank you again and have a great day. Bye everyone.